Nearly 2 decades ago, the urinary microbiome was excluded from the Human Microbiome Project. This is because historically the urine was thought to be devoid of bacteria, and thus it was not the subject of careful microbiological investigation. But over time, and with increasingly precise techniques, it has now become clear that the urinary tract harbors microbiota, and as such, there's considerable interest in characterizing the patterns of bacteria and other microbes in the urine in both the healthy state and disease conditions. Overactive bladder, which is characterized by urinary urgency and frequency and incontinence, it has symptomatic overlap with urinary tract infection. Thus, we hypothesized that microbiota patterns would be associated with the overactive bladder condition, and we designed a clinical study wherein we assessed the urine and clinical factors of 150 patients, about two-thirds of whom had overactive bladder and one-third of whom were healthy controls. We identified key differences in the urinary bacteria between patients with overactive bladder and those without. Specifically, we found that particular strains of lactobacillus, which is a common probiotic species, were more abundant in the healthy control population. Interestingly, the same species were more common in the group of overactive bladder patients who responded versus did not respond to therapy for their overactive bladder. Thus far, we've demonstrated an association, but this finding does not necessarily translate to a causal relationship. So, based on our results, we hypothesize that specific bacteria may help to structure healthy patterns of the microbiota in the bladder, which may protect against an unhealthy state. Future studies and experiments are being designed and performed to test this hypothesis, and in the future, it's possible that augmenting overactive bladder therapy with an oral, intravesical, or vaginal probiotic could improve treatment response. The safety and efficacy of such an approach requires empiric experimentation and trials, and the study we report here paves the way for this future work.